So hello my dear friends, welcome back to another tutorial video that is lactose operon. It is given on the screen, you can see. So our YouTube channel biology class 444, and it is the operon part 2 video. I promised you that I will bring another operon video where I will say about lac operon, and now it's lactose operon. It is written, you can see that. So let's begin. So if you can remember that last video, I told this slide that I showed this slide that how many types of operons are there. So there is three types of operon. Actually, there are several types of operons, but we have to read these three because it is also given in our syllabus, and also we have to study, and also these three are given in our books. So let's begin the lac operon. I told that three types of operons are there: lac operon, tryptophan operon, and arabinose operon. So the topic comes lac operon. So let's see. So here the main carbon source of the lac operon would be the lactose, because it is lac operon. Lac means the lactose operon. So lactose is a disaccharide consisting of two monosaccharide. You can see it is written here that lactose is a disaccharide and consisting of two monosaccharide, that is D-galactose and D-glucose. And here one thing we have to remember, that three proteins are necessary to synthesize the lactose operon, because when the lactose is present, then three proteins are needed. That is one, beta-galactosidase, number two, lactose permease, number three, beta-galactosidase transacetylase. So these three proteins you have to remember again, that beta-galactosidase, lactose permease, and beta-galactosidase transacetylase. So the main thing of this operon is the lactose will be the main carbon source. That should be in our mind, that lactose, when the lactose would be in the carbon source, means that in the growth medium when lactose will be present, this operon model will be worked for that. So here the question occurs, what is beta galactosidase? So lastly I told that beta galactosidase is one type of protein, and among those three, beta galactosidase is one. So there are two types of work you can find out in the whole operon series, that this enzyme can break down the lactose into D-glucose and D-galactose. Lastly I told that that lactose, what is the monosaccharides of lactose, that D-glucose and D-galactose. That's the main thing. The beta-galactosidase will break down the lactose into D-glucose and D-galactose. Here you can see this is the D-galactose model, and this is the glucose model, D-glucose. And another one work of that is it catalyzes the isomerization. That isomerization is the conversion of different from one to another. So it catalyzes that isomerization process to make lactose into allolactose. And there are several functions you will notice in that whole lac operon series, that what is the work of allolactose. You can nextly you can understand when we will further study of it. So now you have to remember two things, that beta galactosidase can break down lactose into two monosaccharides, that galactose and glucose, and it can convert lactose into allolactose. So coming to the next, which are where two proteins are left. Here you can see the lactose permease and the beta galactosidase transacetylase. So lactose permease are also called the M protein, and this is one type of protein found in the E. coli cytoplasmic membrane, actively transport lactose into the cell. So what happens? The lactose permease, this protein can be found in E. coli cytoplasmic membrane, which helps to transport lactose into the cell. So actually what happens, without the help of lactose permease, E. coli bacteria cannot get the lactose from medium into the cell. So lactose permease it is present in the cytoplasmic membrane, which helps to enter lactose through the cytoplasmic membrane into the bacterial cell, means E. coli cell. So what we have to know that without lactose permease, this lac operon will not be held. So coming to the last and not in the list, that is beta galactosidase transacetylase protein. Actually, this protein work, and where it works, it couldn't find out by several scientists. So there are some conspiracy within it. So, but some of them are said that this enzyme transfer an acetyl group from acetyl CoA to beta galactosidase. So we have to remember this because many of the books are saying this. So we have to remember this, that this enzyme transfer an acetyl group from acetyl CoA to beta galactosidase. So coming to the next slide, what is written? So my dear friends, you can see a diagram is representing here E. coli cell. It is the inside portion of E. coli cell, and it is the E. coli cytoplasmic cell membrane. And here the blue color represent the permease protein. Here it is written, you can see the permease protein transport lactose from medium into E. coli. That lastly I told that permease protein helps to transport that lactose from the growth medium inside the cell. So here is the permease protein. So if lactose permease protein is unavailable into the cell membrane, so this growth medium lactose cannot entrance through the cell membrane inside the cell. So permease protein is necessary. So here is the lactose diagram. So this lactose, with the help of beta galactosidase, it will be into two monosaccharide. That one will be the D-galactose, another will be the D-glucose. And here is another lactose. With the help of beta galactosidase, it will be converted into allolactose. So coming to the next slide. So here is the another slide. You can see that growth medium is outside of the cell membrane, and this is the E. coli cell membrane, and inside the two lactose is there. One lactose after the catalyzing of beta galactosidase, one reaction will be there, and another one lactose after the catalyzed by the beta galactosidase number two reaction would be there. It will be coming into the next slide. So let's see. So here you can see that reaction 1 and reaction 2. So reaction 1 says that the beta galactosidase break down the lactose and make D-galactose and D-glucose, and 1 mole of H2O, means water molecule, will be released. And the next reaction 2, shows that the beta galactosidase react with lactose and make it allolactose. That catalysis process I told last slide, if you can remember, that beta galactoside has two work. Number one, the making lactose into two monosaccharide, that D-galactose and D-glucose, and the second one, lactose would be converted into allolactose by beta galactosidase protein or enzyme. So there are some facts we have to keep in our mind very clearly that is necessary to understand the total operon model. So these two are for the lactose operon, and you have to remember these 2 points in exam hall 2, because there should be some question like some uh, situation they will give you that if glucose is present in the medium, then what will be the rate of lactose operon, or glucose and lactose both are present in the medium, then what would be the result of that lac operon or operon. So we have to see these 2 points. Firstly, it says that in wild-type E. coli growing in a growth medium containing glucose, so only a low concentration of each of these three proteins is produced. But in the presence of lactose and the absence of glucose, the amount of each enzyme increase coordinately. So these 2 points want to mean that if in the medium containing glucose in huge quantity. So the so the low concentration of these three proteins, which three proteins? That beta galactosidase, permease, and the transacetylase, those three proteins will be produced in a low concentration because in the medium glucose is present, and if there is no glucose in the medium, only lactose is present in the medium, then the amount of these three proteins will be increased coordinately. So we have to understand why it is happening. In the presence of glucose, the low concentration of three proteins are produced, and in the absence of glucose, in the presence of lactose, the amount of these three proteins are increases. Why it is happening? So there are some points occurs that if glucose is present in the medium, then glucose is a preferred carbon source, and because it can be used directly by the glycolytic pathway to produce energy for the cell, so lactose and other sugar converted to glucose to consume energy. So here if the glucose is present, then there is no need to take lactose, because after taking lactose they have to convert it into the glucose. So if glucose is present in the medium, so there is no use of taking lactose, and so Lac-operon will not be held. So therefore more energy can be obtained for the cell from the glucose than from the other sugar source. So if glucose is present, then they can get more energy than the other sugar source. So that's why the glucose is present, then Lac-operon will be in low concentration or will be stopped. And if lactose is present in the growth medium and glucose is absent, then these three types of protein will be increased in concentration and produce in huge amount of. So here is the lag gene. You can see 5 prime to 3 prime on lag gene, and several sites are there. You can see this is the promoter site, this is the lag I site, this is terminator site, this is promoter, lag operator, lag Z plus, lag Y plus, lag A plus, again terminator, and that's it. So this one is the lac I plus, means lac repressor gene. From here, after transcription, one mRNA will be produced, and that mRNA further it will be lac repressor. And here you can see that lac Z plus, lac Y plus, and lac A plus. After transcription, three protein will be produces: beta galactosidase, permease, and transacetylase. Lac Z is responsible of beta galactosidase production. Lac Y is responsible for the permease production, and lac A plus is responsible for transacetylase production, and these three are called polycistronic mRNA. So lac Z plus, lac Y plus, and lac A plus, these are the structural gene, and others like promoter, lac I, lac operator, these are called regulatory gene. Here you can see that it is written lac I plus is lac repressor gene, lac O plus is the lac operator, and structural genes lac Z plus, lac Y plus, and lac A plus. Regulatory genes are promoter, lac operator. This is the lac operator, and this is the lac I plus, means lac repressor, and etc. So here plus, you can notice that here plus is there. Why it is plus? So plus means that it is not mutated. It is actual gene or wild type gene. Where you can see that in the place of plus, minus is there or negative sign is there. That means that gene which symbol is negative. That means that gene is already mutated. So here plus means the wild type gene which are not mutated. Here, those are not mutated gene. In the last slide, I told that polycistronic mRNA. What is polycistronic mRNA, and which one? That in lac Z and lac Y and lac A, from those three genes, that three proteins are produced or one mRNA produced. Those three protein are beta galactosidase, permease, and transacetylase. Those three are called polycystonic mRNA. Why? Because those three are transcribed onto a single mRNA molecule. Means in a single mRNA molecule, those three protein will be produced. So in a single mRNA, those three are present. So it is called polycystonic mRNA. So now question occurs, how many types of regulation are in the Lac-operon? Means how many types of regulation are there in the Lac-operon? So if you are new, so there will be some problem, and you if you have any problem, then do some comment. I will make you understand those problems. So coming to the next slide, how many types of regulation are there in the Lac-operon? So there are two types of regulation. Naturally, we can see that negative regulation or negative control, or positive control or catabolic responses, repressions. Positive control and negative control. So, what is negative control? So all of us we know that the lac repressor bind to the operator site, and the DNA sequence covered by the repressor protein overlaps the DNA sequence recognized by the DNA polymerase. Therefore, when the repressor is bound to the operator, RNA polymerase cannot bind to the promoter, and transcription cannot occur. Because of this, the Lac-operon is said to be under negative control. That means we know that RNA polymerase will bind in a RNA polymerase binding site, and from there the further transcription will happen. Lac Z will produce beta galactosidase, Lac Y produce permease, and Lac A produce transacetylase. But in the negative control, it cannot be happen. Why? Because the repressor will bind in that DNA polymerase site, so that the lac repressor will bind in that RNA polymerase site, so that RNA polymerase are unable to bind with the DNA sequence, because in that DNA sequence where RNA polymerase binds, they are already lac repressor bind. So RNA polymerase is unable to bind, so the negative control happens. So let's see that model. So here you can see that lac I is there, lac I, and promoter, operator, terminator, lac Z plus, lac Y plus, lac A plus. So these are called that structural genes, and others are the regulatory genes. Means they will regulate the total process. So lastly, I told that lac I plus produces after transcription one mRNA, which later produces lac repressor, and these three produces the structural genes, beta galactosidase, transacetylase, and permease. So here what happened in the negative regulation of Lac-operon, when lac repressor here lac repressor will bind in the operator site. So when lac repressor will bind to the operator site, then RNA polymerase cannot bind, because already lac repressor bind in that site, in operator site. So RNA polymerase is unable to bind in this particular site. So when RNA polymerase cannot bind, then they cannot code these three gene. So no structural gene will be expressed. So the point occurs in the negative regulation. The lac repressor gene will sit in this operator site, so that RNA polymerase cannot sit here. So these three structural gene will not be expressed. But here one thing we have to remember, that if lac repressor bind in this site, then the Lac-operon will be stopped. But sometimes it is seen that Lac-operon is not stopped, because why do you know? Because repressor do not just bind and stay, they bind and dissociate. In that split second, after one repressor unbind and before another binds, then and there that RNA polymerase makes some proteins from this structural gene. So what does it mean? That Lac repressor, once it binds, it will not stay for long. Sometimes after it will dissociate and again another repressor will bind here. So in the meantime, when it dissociate and another new repressor will bind, in that meantime, the RNA polymerase bind here and make some structure and make some protein from these structural genes. So when the negative regulation happens in the Lac operon, then not totally Lac operon is stopped, means not totally structural gene expression is stopped. Sometimes it makes the structural genes to produce the three proteins, the beta galactosidase, permease, and transacetylase. So in exam hall, some question comes that during the negative regulation, the three protein or enzyme production will be stopped or not, or total lack operon will be off or on. So the answer will be it will be not completely off, because some a low level of structural gene will make those three proteins, because the repressor do not just bind and stay, they bind and dissociate, and that split second after one repressor unbind and before another bind, the RNA polymerase make from LAC-Z to beta galactosidase, LAC-Y to permease, and LAC-A to transacetylase. So these fundamental we have to remember. In the previous slide, we see that in the absence of lactose, in the presence of glucose in the medium, the negative control will be round. So another type of operant model are there. In the presence of lactose, then what will happen? So in the presence of lactose and in the absence of glucose, some wild type E. coli grows in the presence of lactose as the sole carbon source. So some lactose is converted by beta galactosidase into allolactose. So, so there is the lac repressor, and with the help of lactose, with the help of beta galactosidase, and lactose will be converted into allolactose. And these are the allolactose. You can see that inducer molecule allolactose. This allolactose will bind to the Lac repressor. Here you can see these are the Lac repressor, and this allolactose will bind to the Lac repressor. So this shape change, so you can see the shape are changed. So the shape change is called allosteric shift. So this is the allosteric shift of Lac repressor. So Lac repressor, after binding with allolactose, it will change its shapes, and then the name will be given allosteric shifts. So for that, the repressor lose its affinity for Lac operator and dissociate from the site. So what will happen? So when the allolactose will bind to the Lac repressor, it cannot bind to the Lac operator site. Obviously, if the shape will be changed, so they cannot sit here. So the free repressor proteins are also be altered by binding to the allolactose. So no Lac repressor will be there. So the RNA, um, RNA polymerase can bind to the Lac operator site, and then the transcription will be happen. Transcription will happen, and then three proteins will be produced by these three genes, and the RNA polymerase will encode these three genes. Then the three proteins will be produced on mRNA, and that is from LAC-Z beta galactosidase gene, LAC-Y permease, and LAC-A transacetylase. So this is the another type of operon. It will happen in the presence of lactose and the absence of glucose. So lactose present as a sole carbon in wild type E. coli, and the lactose became allolactose with the help of beta galactosidase gene that I told. The presence of lactose will be converted by beta galactosidase into allolactose, which will bind with Lac repressor, and it will change the Lac repressor shape, and it will be called allosteric shift. And then for the changing of their shape, they cannot sit in Lac operator site. Then RNA polymerase will take the advantage of it, and RNA polymerase will sit here and encode these three enzymes, and these three protein will be produced: beta galactosidase, spermiase, transacetylase after transcription. Got it? I think you can remember this slide that I showed you before, that how many types of regulation are there in Lac-operon. So lastly, I told that negative control that I told before this slide. So now left is the positive control. So the positive control and catabolic repressions are left. So let's see what is that. So positive control and catabolic repressions. What is that? It is also one type of Lac-operon model. Several years after Jacob and Monod proposed their operon model, researchers found a positive control system that also regulates the Lac-operon. So after the research of Jacob and Monod, and they proposed the model of operon model, that negative control they proposed. So after that, several years later, some researcher got that another one positive control system that also regulate the Lac operon. It's a system that function to turn on the expression of the operon. So in negative control, what we saw that in negative control, total Lac operon process is totally stopped, or a little bit operon will be there. But in this system, the total function is turned on, and the Lac operon will be again restored. So the system ensures that the Lac operon will be expressed at a high level only if lactose is the sole carbon source, and not if glucose is present as well. So lastly, I told that glucose effect, that you can remember that few slides ago I told that what is glucose effect. So here also you have to remember that glucose effect. So if lactose is present in high level and the absence of glucose, the positive control will be held. So in the presence of lactose, this positive control will be held. In the high quantity of presence of lactose, these control are seen by some researcher, and at that point, glucose should not be present in that media. So let's see the figure of positive regulation. So here you can see a protein called CAP, catabolic activator protein, and another was CAMP, cyclic AMP or cyclic adenosine 3' 5' monophosphate. Both of them will bind and make CAP, CAMP active or CAMP complex. So here you can see CAP, catabolic activator protein, it is a protein, and another 4 CAMP, cyclic AMP, uh, will bind with each other and they will make a complex. Here you can see this is a cap camp complex, and this is called the positive regulator molecule because it will regulate the positive regulation or positive control. So here you can see a gene. What is the difference between previous those slides gene and this one? One vital difference is there. You can see that before promoter and after Lac I, here is a cap site. Here the cap and camp complex will sit. So it is the upstream of promoter, and then what will happen? Let's see. So here you can see the cap camp bond binds to cap site adjacent to the Lac promoter. Means beside the Lac promoter cap site, the cap and camp complex will be placed. It will sit here. So same thing is there. You can see this one and this one same. The cap site binds by the cap camp complex. So after the cap camp complex bind to the cap site, RNA polymerase hollow enzyme will bind to the promoter. So what will happen? the after binding of cap complex to the cap site, they will bind RNA polymerase holoenzyme to the promoter site so that the transcription will be begin. So this RNA polymerase will go towards the 3 prime end, means it will go front portion, means Lac operator, Lac Z, Lac Y, Lac A. So here you can see the transcription begins. The RNA polymerase went towards the 3 prime end, and transcription and translation happened. mRNA produced. Here you can see it is a mRNA, 5 prime mRNA. mRNA produced by the RNA polymerase holoenzyme, which encoded the Lac Z, Lac Y, and Lac A. So positive control or positive regulation is this one. You have to remember that cap and camp complex will be there, and this is the vital, and this plays an important role as a positive regulator molecule. Without this, RNA polymerase holoenzyme cannot bind to the promoter site, and then mRNA transcription and translation will not be held, and the cap complex will bind to the cap site. And where is the cap site? Upstream of the promoter and after Lac I. So here is the cap site. So coming to the next slide we will explain some notes that what would be the, these are the vital notes we have to remember again and again for the positive control. If the glucose effect happened in the positive, in this positive control, the Lac-operon will be expressed at only very low level. Even the lactose is present in the medium. So same thing we have to remember that in last negative control also we saw what is glucose effect. So if glucose effect happened in here also, then the Lac-operon will be expressed at only low level. If the lactose is also present in high quantity or low quantity in the medium, but if glucose is present, then only a very low level of Lac-operon will be expressed. Why it is happening here? Because the glucose reduces the amount of CAMP in the huge quantity. So we know that CAMP, if it reduces, then the cap-camp complex will not be held. So if the cap-camp complex will not held, then the RNA polymerase cannot bind to the Lac operator site. So insufficient of cap-camp complex present causes no requirement or recruitment of RNA polymerase to the Lac promoter. So obviously if the camp is reduces, so the insufficient of cap-camp complex will be produced. So insufficient of cap-camp complex causes the no recruitment of RNA polymerase. So in the Lac promoter site. So the very low level of Lac-operon will be expressed, or Lac-operon will be turned off. So if you can remember that in negative control, the repressor seat, the repressor model is there, that if the repressor are removed from that operator by the presence of allolactose, then the Lac-operon started. So here it will not work. If the repressor are removed also from that operator by the presence of lactose, then also it will not then also positive control will not be held. Why? Because the glucose will be present, and the glucose will causes the insufficient amount of camp. So insufficient of amount of camp causes the insufficient of camp complex. So RNA polymerase then also cannot see to the left promoter side. So the repressor are removed also from the operator by the presence of allolactose. Then also Lac-operon will be expressed at only very low level. Why? Because RNA polymerase cannot bind without the help of cap complex. So this we have to remember. that CAP-CAM complex is the vital for the positive control, because CAP-CAM complex is the positive regulator molecule for the positive control or positive repression. So, yeah, insufficient amount of CAP-CAM complex causes the low level of Lac-operon expressions. So thanks to everyone. Wait for the Operon part 3, where I will describe about tryptophan operon or TRP operon, and follow our channel for the further videos. So if you like the video, please like and subscribe for the upcoming Operon part 3. So if you have any problem, do some comment. I will try to make you understand. And if you want to share your knowledge, please do some comments. Thank you, and follow our YouTube channel Biology Class 444. Wait for the Operon part 3. Bye bye.